Okay, so in the first part, we covered just basic information about epithelial tissues and basic ways to go about naming them. So if you recall, we talked about the cell shape and we talked about the cell layers. So you can be flat, cube, or columnar, or you can be squamous, cuboidal, columnar, or you could be simple or stratified. You could also be pseudostratified, right? So if you haven't watched that video, watch that video first. So in this part, in this video, what we're going to look at are specific examples of each of those. Okay, so on Histology Guide, you can scroll down and go to different sections that have examples of these different types, right? So if we look at something like the eye is going to be, in terms of the cornea, the outer surface of the eye, this is going to be composed of a simple squamous epithelium. Very thin, because we need light to be able to pass through the cornea as it's coming into our eye to be refracted and focused on our retina. So here you can see, if we zoom out and give you some perspective, here's our cornea, right? And if we look here, we can see this inner layer here of a simple columnar, a simple squamous epithelium on this inner surface of the retina. So you see lots of individual cells. Every black purple dot that you see, that is representing a nucleus of one of these cells. Okay, so everywhere you see a dot, this is a cell. It's a nucleus, but it's representing one cell. And we see that most of these are very flat in appearance. They're this flattened out, squamous appearing cell, right? So this is a very good example of simple squamous. We can also find them here in the kidney associated with the glomerulus. We looked at this in the first part as well. So circling the outside of this inner ball of capillaries known as the glomerulus. This is called the glomerular capsule on the outside. We have this nice thin section of simple squamous epithelium. If I go back and I go down to respiratory system, where is respiratory? We can look at our alveoli, and we can see that we have a very thin, simple squamous tissue that is associated with our alveoli. So if I come over here and we look at an example of something like, we come over here. So in our lungs, we have alveoli where we have gas exchange occur, right? And so this process of gas exchange requires a very thin membrane in order for the gas to diffuse across this barrier. So we've got to go from the inside, so this white region here, from this region across this membrane into the blood, right? Well, one way to accomplish this is to have a very thin membrane that allows the gas to diffuse very easily across. If you had a very thick membrane, this would make the diffusion much more difficult, all right? So if we go back to epithelium, you can look at some other examples here, and remember, come over, look at the inside of a blood vessel. You have the inner lumen, this is where the blood would actually be circulating, and lining the inside of this is our epithelium, sometimes called the endothelium, with our blood vessels. And this is an example of a simple squamous epithelial tissue, right? So if we go and now we look at simple cuboidal, you find simple cuboidal in areas like the ovary. You can find them associated with the kidney. You can find them associated with the thyroid gland as well. And cuboidal epithelial tissues are generally going to be involved in some type of absorption and secretion process. Okay, so if we zoom in and look here, it should be very easy to identify this as simple cuboidal. Again, every purple dot is representing a nucleus, and so you can kind of trace around one individual nucleus and get a roughly cube-shaped cell. You also see lots of simple cuboidal epithelial tissues in the kidney with our renal tubules. So all of these, you kind of see these large circular regions. Every one of these is a glomerulus, and then everything in between, you can see these little openings. All of these are composed of simple cuboidal epithelial tissues. The reason for simple cuboidal, as we said earlier, that it is involved with absorption and secretion, and one of the primary functions of our kidney is the reabsorption and secretion of various things into and out of the blood, right? As we're wanting to reabsorb things or ultimately excrete things. So if we zoom in here, you can see very nicely along a lot of these tubules, it's nothing but simple cuboidal epithelial tissue. So you have these long tubules here. You have regions called proximal tubules. You have other regions called distal tubules. And all of these, no matter where you go in the kidney, these are all going to be made up of a very nice, simple cuboidal epithelium. If we go and we look at something like thyroid, so in our endocrine system, and we go and we look at something like the thyroid gland, where's our thyroid? And we zoom in, we should be able to see a nice simple cuboidal surrounding our thyroid follicle. So every one of these large circular regions is a follicle, a thyroid follicle, and you can see surrounding the thyroid follicle, we have lots and lots of cube-shaped cells. These are just a single layer, so we call these simple cuboidal epithelia, right? And so with the thyroid, obviously, we're involved with the absorption of iodine to ultimately make our thyroid hormone, which our thyroid hormone is then secreted out into the blood to exert its metabolic effect on the body, right? So if we go back and we move down now to simple columnar, right? Simple columnar is a very common epithelial tissue to find associated with our digestive tract, okay? So here you can see all of these cells. Again, every kind of dark black purple dot is representing a nucleus. And if you follow these cells, you can kind of make the outline of some of these. And these are roughly rectangular and kind of column shaped. Now, not every one is going to be. Some may be kind of flattened or squished by nearby cells, but the vast majority of these are going to be roughly column shaped. Okay. Now, you may notice on the surface, you kind of have this little border running on the surface. This is sometimes called the brush border. All right. And what this brush border is, it is essentially microvilli. Okay. And so microvilli are these small little extensions of the cell's plasma membrane on its apical surface. So we're called the apical surface is that which is open to the lumen. So in this case, the white region here is the lumen. And these microvilli are in contact with the lumen. And so we said in the first part that these apical extensions called microvilli serve to increase the surface area for this particular cell, for this particular epithelial tissue, to absorb more and more things. So if this is associated with the GI tract, one of the main functions of our GI tract, along with breaking things down, is to absorb things, right? We need to absorb it and get it into the blood, whether it's sodium or it's water or it's glucose or it's amino acids, whatever it may be. We have things that are able to break down those products, and then with the assistance of the microvilli, we're able to absorb those things into our gut, right? Now, we can also find these associated with the ovary. So if we come here and we look at simple columnar and you zoom in, you see a nice simple columnar. Again, dark purple is nucleus, and make out the outline of individual cells. It's roughly column shaped. Now with these, you notice something different, or hopefully you notice something different. This is not microvilli, this is cilia, okay? So in the first part, we said that cilia are involved with the movement of things, movement of something over the surface, over the apical surface of that epithelial tissue. So in this case, if we're in the uterine tube here, or what's sometimes called the fallopian tube or the oviduct, we're moving the ovulated ovum, the ovulated egg, over the surface of these cells as it makes its way down the fallopian tube to where it may ultimately encounter and be fertilized by a male sperm cell, and then it can go to the uterus and it can implant, right? So in this case, we have simple columnar, not with microvilli, but we have it with cilia to help propel and move the ovulated egg over its surface, okay? All right, so if we come here now and we look at our pseudostratified, one of the best places to look for pseudostratified columnar epithelia is in the respiratory tract. So if we zoom in here, we can see, and now we're kind of inverted here. So this is the basal layer down here, and this is the apical layer down here, right? And so we said in the first part that the way you define a simple epithelial tissue is that all the cells touch the basement membrane, okay? For this tissue, this is technically a simple tissue, okay? All the cells touch the basement membrane. However, not all of the cells reach the surface, okay? Reach the apical surface. They all touch the basal membrane, the basement membrane, but not all of them reach the surface. So what this means is that you have this kind of appearance that the cells are stacked on top of each other when in reality, that's not the case. And so because you get this kind of false impression that the cells are stacked, you get nuclei that appear at different levels in the tissue. So you have some nuclei that are very superiorly located in the tissue near the apical surface. You have other nuclei that are at the very bottom. You have ones that are in the middle. So you can see that all these purple nuclei are scattered all throughout the epithelial tissue. This is one key indicator to help you recognize this as pseudostratified columnar and not simple columnar. All right. So keep this picture in mind with the nuclei at different levels, and I'm now going to show you simple columnar again, right? And so simple columnar, when we came in here, the nuclei are all in a row. So if you look at this section here, all of these nuclei are perfectly in a row, more or less perfectly. If we go back to our oviduct example and you look at the nuclei are more or less all lined up in a row. They're not scattered throughout, okay? Now, if we look back at our pseudostratified, we also see that it has an apical specialization, and in this case, it's going to be cilia. So we said that the simple columnar epithelial tissue we just looked at had cilia to help move that ovulated egg throughout the uterine tube. In this case, we have cilia because we're going to be moving mucus over the surface of the epithelial tissue in the trachea and help move it up toward the pharynx, up toward the oral cavity, where you can then do one of two things. You can swallow it or you can choose to spit it out, right? So in this case, the mucus is going to serve to trap dust and debris and foreign cells and pathogens and bacteria that we inhale, and then we can work those things up and we can get rid of them in the body. So this kind of border that you see here, this is not microvilli, but this is cilia on the apical surface, all right? Okay, now if we go and we start looking at true stratified tissue, so true tissues that have more than one layer, we'll look at a couple of examples, primarily two. We'll look at stratified squamous, and then we'll look at something called urothelium, sometimes called transitional epithelium, all right? Now, our stratified squamous, so think of the word stratified squamous. We have stratified, and we have squamous. So stratified is telling us we have more than one layer, so at least two, usually more than two. And then squamous, we have cells that are roughly flat, okay? Now, stratified squamous, you would think, based on the name, that all the cells would be perfectly flat. That's not exactly the case. The cells near the surface, the apical surface, are relatively flat and squamous shaped, but the cells at the basement layer, the basal layer, are more kind of rounded or cube shaped. Okay, so keep that in mind. So if we look first at what's called keratinized stratified squamous, this is what we find in our skin. So if you look down right now and you look at your hand, you look down now and you look at your arm, you look at your leg, you look at your feet, this is the skin, or this is the tissue that you have on that skin. The keratinized comes from this outer tissue here, which is called keratin. It is a type of protein, and it layers and is sitting on top of the skin, and it serves to protect it. It serves somewhat of a kind of waterproofing, water-resistant type function. Okay. Underneath, we now see our epithelial tissue. In this case, this is a stratified squamous. Now, when you look at these cells, they don't necessarily look perfectly flat. They don't necessarily look squamous. But if you look at something like this, and we zoom in, and we go to the top, now we have cells that appear to be more flat and more squamous-like, right? So in this case, this is an example of non-keratinized stratified squamous epithelium. So when you're talking about stratified squamous, you have to differentiate between, is it keratinized, is it non-keratinized or unkeratinized? This unkeratinized, this is where you find this in things like the inside of your mouth. So you run the tongue on the inside of your cheek, on the inside of your mouth. That's a non-keratinized stratified squamous epithelium. The food that runs down your esophagus and touches the inside of your esophagus as you're swallowing, that's in contact with a non-keratinized stratified squamous. The inner lining of the anus and the vagina are both also composed of this non-keratinized stratified squamous. Okay, you don't need the keratin in this case because it's more deep in the body. It's more within. It's more internal, and so the risk of it becoming dry and becoming brittle with a lack of moisture, it's not as much of a risk. So you can see here, it's kept constantly moist, and that's because it's located deeper within the body, whereas our keratinized epithelium that's on the surface of our skin, you can experience drying out of the skin much more easily than you can with things like the inside of the mouth or the esophagus or things like that. Okay, so when you're defining and looking at stratified squamous tissues, remember to differentiate between keratinized or non-keratinized. Okay, now when we get to our skin lecture, and we look at skin again, we'll see that this is in one type of skin. This is what's called thin skin. We'll also see a section, what is called thick skin, and it will look a little bit different than this. Okay, all right, and then lastly, we're going to look at transitional epithelium. So we're going to skip stratified cuboidal. So transitional epithelium, the term transitional gives the impression that this tissue can change. It can transition in shape, okay? And that's exactly what we see. And when you think about its location, it makes perfect sense, right? So this is a beautiful example of anatomy dictating physiology or structure dictating function, all right? So because of the shape and because of the appearance of this transitional epithelium, it's going to behave like something else, right? So transitional epithelium means that the cells are going to transition in shape from the basal layer to the apical layer. So if you look here, on this apical layer, you have these large, round, what are called umbrella cells, okay? So if we click here and we look at these umbrella cells, these are these large, round ones that you see on top. Sometimes they can be binucleated. They can have two nuclei. On the underside, you see that and going closer to the basal surface, you see the cells kind of begin to change shape. Some of them kind of look maybe columnar in shape. Some of them look maybe kind of cubed and kind of boxy shaped, right? So what this means is that as the bladder fills with fluid, right? So as it fills with urine during the process of urine formation, it's going to stretch out this epithelium. And because the cells are arranged in this manner, it allows the expansion of this epithelial tissue to accommodate this increase in fluid volume, okay? So as that happens throughout the entire inner lumen of the bladder. So, right, if we look this inner white space, this is the lumen. So this is where the urine would form. You see you have these large folds in the bladder called rugae. So that helps with the expansion of the bladder. You also have this epithelial tissue that we're looking specifically at, in which case the cells are going to change shape. And as you fill with fluid, it allows the bladder to expand very, very nicely. Okay, so you find this in the bladder. You can also find this in areas like the parts of the ureter, and you can also find it in parts of the urethra as well. So if you look at this image here, you can see you have these large umbrella cells on the surface, and then as you go down, the cells become more and more kind of flat and squamous-like, okay? But they really kind of more stay cuboidal, okay? So this slide here shows a good example between relaxed transitional epithelium, so in this case, versus extended. So this means in this tissue here, this is when the bladder is full. So you can see these cells look very stretched. They look very flattened out as the bladder would be full of urine. And in this instance, they look a little bit more rounded and kind of plump, right? So in this case, the bladder would be empty or would not have as much urine in it as the previous ones, right? So here you can see the dome shape of the umbrella cells. They're very kind of round and dome shaped. In this example, the umbrella cells have become kind of flattened out. They're not quite squamous, but they're just less round, less dome shaped than when you have a relaxed bladder, okay? All right, so that covers all of our different epithelial tissues. Again, when you're thinking about these tissues, remember to look at multiple different images of the same thing, right? So when you're looking at simple cuboidal, look at 3 or 4 or 5 different images of simple cuboidal. Same thing for all the other ones. Look at the number of layers, look at the shapes of the cell, and try to think, okay, because it looks like this, because I see one layer of very flat cells, what is this tissue going to behave like? Or what is its function going to be? So you begin to relate this anatomy to this physiology, this structure to this function, okay? And that should help you as you learn these different types of tissues.